Hi, good morning, everyone. Um, Welcome, Dr. Mohit. Um, it's 9:55 a.m. here, so we can wait another few minutes. Few other people may join, and or if you want, you can start also. It's up to you, totally. Uh, you can hear us, right? Hi, Dr. Mohit. Can you speak?
Good morning. It's Veronica Byers. Thanks for having this.
Hi, good morning. Um, let's just hi, Dr. Mohit. Can you hear us clearly?
Yes, clearly.
Oh, okay, okay. I think Dr. Mohit is downloading something. Okay, okay. We can just wait a little bit for him. Yes, now we can hear you.
You can hear me?
Yes, yes. Now we can hear you.
Okay. I'll share my slides.
Yeah, sure.
Screen. Can you allow the permission to share the content? Abu?
Yes, just give me 1 second. I think
Can this be shared to make sure you allow the permission to share content?
I think the permissions is already given.
There.
Yeah, yeah.
I will check. I will check.
Good morning, everyone. Those who joined a little bit late. Good morning, Dr. Arnab. We are just waiting.
Waiting.
Take your time. No worries.
Or else what
Just
I'll do is I will check. I will check with the app. Okay.
okay. Sure,
Uh,
sure, sure.
I downloaded the app. I'm now opening through the Google Chrome,
Yeah, yeah,
but
yeah.
it's showing we can't display your shared content. Make sure that you have allowed permission to share content, and then try again. It's showing. I'll do one thing. I'm logging out and I'm joining with the WebEx app. Okay.
Okay, sure, sure. No problem.
Okay. 1 minute.
Good. Well, we can see your,
Yeah,
yeah. Yes, we can see your screen. Hello, can you hear us, Dr. Mohit? You can hear us, right, Dr. Mohit?
can I join?
Yes, yes. No, no, no. You can start. Yeah.
Yes, yes. I can hear you.
Yes, we
Can you
can
hear
hear
me?
you also. It's just taking little
You can
bit of
hear
time,
me?
I think.
Yes. Hear me, right?
Yes, I can hear
You
you,
can hear me, right?
but it is little bit broken.
Hello. Okay. So I can start now.
Uh, yes, you can start now.
So I can start.
Yes.
It's getting broken
Yes, sound is little bit broken.
now.
Now we can hear you.
Okay. morning, good evening, good afternoon, wherever you are. Present. I'm working as a Habib. It's getting on, or
I think there is some technical difficulties. Let me uh, let me figure it out. Just wait a little bit. I think we're gonna face this kind of yes, Dr. Mohit. I think you left and somehow uh,
can you hear me now?
yes, I can hear you now.
Okay, okay. There's problem with the mic of my laptop, I think. Okay, I just want to share my slides so you can see the slides.
Yes, we can see the slide.
So it's properly audible.
Yes, it's properly audible. We can hear you also.
Okay, okay. Hello, hi everyone. This is Dr. Abdul Mueed. Presently, I'm working as a clinical scientist in Saudi Arabia in a hospital called Dr. Suleiman Al Habib Medical Group. Uh, just everyone is preparing for SMB or MB for molecular biology, and I just want to share my little bit experience of NGS. Actually, I have not much have wet lab experience of NGS, but I have more experience in analysis, interpretation parts. So there is analysis like whole exome sequence analysis. So I'll talk a little bit about the wet lab work also, as well as analysis part. Okay. So here you can see the slide shows different organelles of the cell. This is the basics, structure of the organelles, where you can see here uh, uh, the chromosomes are inside the nucleus, and as you know, for to extract the DNA from this nucleus, you should break down the cell, then the nucleus, and purify this genetic material, DNA. So it's all depend on what your experiment is about. You want to extract DNA or RNA, for example, to check for example, for sequencing of RNA. So you should extract the whole RNA, means mRNA, RNA, tRNA. If you want to extract only DNA, so you will extract the DNA. So the procedure will be different. So I'm not talking about more on the procedure of extraction. I'm more talking about the NGS. Okay. So what is DNA sequencing? So DNA sequencing is to determine the number and order of nucleotides that makes up given molecule of a DNA. So why we have to do this DNA sequence? This is my first question. So anybody can tell me why you want to do DNA sequencing?
Maybe to understand any kind of disease or mutations, uh, that could be one
Okay.
reason.
Okay. To study of what?
To study of to understand the sequencing of a gene or understanding the disease causing mutation.
Mutation, but that mutation you can do by just doing by PCR, RFLP, or by real time based PCR, like targeted based PCR, to just check mutation profile, specific target doing by PCR, like PCR, real time based PCR, or restriction, or what is called AFLP, RFLP, just by PCR, RFLP. That also you can do by checking to know the mutation, specific mutation. But why we do DNA sequencing? To know the sequence of each nucleotide. If you, for example, if you want to sequence a gene, so you want to know the sequence of that gene. So that, for that reason, we need to do DNA sequencing, right? So, or else if you want to do the targeted mutations, like for example, to know the what is the exact KRAS mutation, for example, KRAS codon 12 or codon 13 of the from the cancer cancerous tissue. So you, that, for that you can do directly, you can do the targeted mutation screening, like RFLP, real-time based PCR, or specific targeted panels. But for sequencing, if we should know the specific nucleotide sequence of a targeted region or targeted panels or whole exome sequencing. Okay, this is a briefly I want to tell about the quick history of sequencing. As at 1869, there's a discovery of a DNA. Then there will be chemical characterization of the DNA. Then structure of DNA resolved at 1953 by Watson and Crick. Then at 1977, the sequencing by synthesis and sequencing by deletion has been started. The first automated sequencing machine, that is actually Sanger sequencing, started in 1986 or 1980, and they started Human Genome Project. Okay, and I will tell the what this is the cost and first sequencing of bacterial genome has been started established in 1985 by Sanger sequencing. Okay, and then NIH, National Human Genome Project, and Celera company, they started the Human Genome Project, and it cost, that time it cost around $3 billion. The duration of the project was 13 years. And first generation sequencing has started 2005, and in 2013 there is a database called NCBI database. So around 10,000 genes has been put up into the database. Now you have lots of database like Exact, Genomad. Exact is exome database, and exome whole exome sequencing database, and Genomad is whole genome sequencing database, that I will tell you later in my other slides. So this is the this is the history of when the Sanger PCR invented, DNA micro array invented. So let's in previous 1995. So Sanger is the first generation sequencer. Now also we use Sanger sequencing. I will tell you in which application we use Sanger sequencing. Then capital electrophoresis has started in 1998 or 2000. 1998 or 2000, capital electrophoresis has started. Then second generation short reach generation sequencing and single molecule sequencing started in 2015. So these first generation, second generation, and third generation sequencing, now there is fourth generation sequencer like SMART, that is from Pacific Bioscience, that is fourth generation, or from Proton, that is fourth generation sequencer. Third generation sequencer are from Illumina or from Ion Torrent. So first I will tell you about Sanger sequencer. So what is the main difference between Sanger sequencing and next generation sequencing? Anybody can tell?
One difference we can tell is like maybe Sanger sequencing can do very short amount of sequencing, but next generation sequencing can do little bit long, and also like different kind of samples they can do at a time. Like for like Illumina can do a lot of samples at a time.
Yes. So Sanger sequencing, but it is a gold standard sequencing. Okay, and it's a gold standard means uh, uh, to check NGS, when you do NGS and if you find mutations in X gene or in any gene, if you find mutation, but to confirm the mutation is right or is present or there or not, you have to do Sanger sequencing. Okay, because this is the ultimate confirmation method, or we can say validation method from the NGS. So if you sequence a case like from pediatric case or a child having a disease, so to confirm that mutation, you should do Sanger sequencing. Okay, and Sanger sequencing we can do a short region, as Mr. Abu Bakr Siddiq said. We can sequence short region of a exome or genome. Okay, but for NGS you can sequence all the region of exome sequencing. Okay, and so in Sanger sequencing, for what we anybody has done Sanger sequencing in the presentee? Anybody has done Sanger sequencing? Adil, Ahmad, Ghazi, Amna, Miaz, Olivia, Sana, Sajid, Veronica. So anyone has done Sanger sequencing in your lab?
I did not do
Okay.
personally, but I just saw like different departments did that, so I saw them
Okay.
doing in real life, but I did not do personally. So,
Okay. To do Sanger sequencing, first you should do PCR. You should do normal PCR, like how you do PCR. You should design the primers. So you know how to design the primer, right? So you should design forward and reverse primer, and in AC, because I have taken AC, they ask about the primers and what is the conditions required, annealing temperature, TM calculation, those things they will ask in the ASCP. Okay, and first you have to design the primer for the specific region, for specific target region. For example, if you want to design a primer for a BRCA1 gene, okay, BRCA1 gene, and which is specific exon. So you design a primer for this specific exon, like for example, exon 1 or exon 2. So we design a overlapping primers with the targeted sequencing for the targeted intron region, forward primer, and reverse primer also in intron region. So you cover the coding region of the exon, because you look the coding region of the exon, right? So you first you design the primer, then you do the PCR. So here you can see the primer replication, so how the paired DNA sequence bind, and here parental strand melting, first cycle, second cycle. First you will have the denaturation, annealing, and extension. So what this the PCR, you will have same thing in Sanger. First you should do PCR. After checking the PCR, then after amplification you will do, next thing is we call cycle sequencing. In cycle sequencing, what we use is we use DDNTPs. Okay, that is called dideoxynucleic. So what is the dideoxynucleic has? Anybody knows what is the difference between DNTPs and DDNTPs?
it will miss one of the oxygen. That's why like the next
It's
next
letting the
phase will not come.
hydroxyl. It's
think
Yeah,
letting the
Adil,
hydroxyl ion. So
yeah,
it
at
can
third,
terminate the reaction.
yeah, at 3 prime. So any polymerization should occur, it required OH, 3 prime OH group. Okay, it requires 3 prime OH group. Okay, in the DDNTPs, it doesn't have the 3 prime OH group. So I'll go to the next slides. See in here you can see here you will not have 3 prime OH group. Okay, so each nucleotide DDNTPs has different fluorescence. Previously they were using labeled P32, that is phosphorus labeled P32 labeled DDNTPs. But now nobody is using this because this is outdated and this is carcinogenic. So they use phosphorus P32. Okay, so presently we are using dideoxynucleotide DDNTPs, and each DDNTPs like guanine, adenine, thymine, cytosine is colored with different dye. So if you see here you can see the electrophotogram. This is the electrophotogram. So when you do Sanger sequencing, you will get like this peak. Okay, and this is the actually outdated gel, like electrophoresis. They used to run by gel before, 20 years before, and then they used to read different nucleotide by this bands A, T, G, C. Okay. Then you will have presently now you will have the Sanger sequencing like this peaks. So this red called T and blue is C, G is black, and A color is adenine. Thymine is T. Okay, that is red color. So when there is so if you have this template DNA sequence and you design a primer, so when you do cycle sequencing, you will add only one primer. You can't add two primer at a time. For PCR, general PCR, you will add forward and reverse primer. But for Sanger sequencing, dye-terminated sequencing, you will add single primer, either forward or either reverse. Okay. So in lab, what we do is we set a PCR. We'll add PCR product. Okay, it is purified PCR product. So we purified, and that and for now presently we using purification by what purification we do by PCR? Anybody knows? PCR purification. This thing is there in Lila book. So what we do is PCR. We do PCR by cleanup, that is by exosap. We use exosap. So to remove the DNTPs, to remove the primers, leftover primers, we use the exosap. And after exosap, we add the DDNTPs and other buffer enzymes to carry out the cycle sequencing. So here you can see in the template there is an extension from 5 to 3 prime extension. Then there is incorporation of DDN. If there is incorporation of DDNTPs, so there will be terminations. Okay, and the termination read by the camera. So here you can see. Here you can see the gel, and there's electrophotogram on the right hand side. Here you can see each nucleotide is read by the camera AAT. So if there is any DDNTP incorporation, so the camera will detect. So each sequence it will detect. Okay, and for Sanger sequencing, maximum length you can achieve 750 to 800 base pair. You can't achieve more than 1000 base pair. So when you're designing a PCR primer, so you should target maximum to check 600 to 700 base pair, and always you should be take in mind when you design a primer, the first read in the sequencing, Sanger sequencing, the quality wise, the first 10 to 15 sequencing, the quality wise is not good. The first 15 and the last 15, the first 15 and last 15, the quality will be not good, because starting the sequence, Sanger sequencing, quality will be not good. So in between, the quality will be good. Okay. So now you can see there is a DNA sequencer called ABI 3130-3700. So this is 3730 capillary. So you will have now presently you will have different types of automated DNA sequencer, that is Sanger sequencer. Okay, so I will show you the next slide. So this is the presently Thermo Fisher Scientific. Okay, applied biopsy Thermo Fisher Scientific. They have different Sanger sequencer. This is 4 capillaries. This is 8 or 24 capillaries. This is 24 capillaries, and this is 96 capillaries. So you have different Sanger sequencer, 3500, 3730, and this is a polymer. Polymer is like a gel. Okay, to separate this nucleotide in the capillary gel electrophoresis. Okay, so in this Sanger sequencer, you can do Sanger sequencing and you can do fragmented analysis, DNA fragmented analysis. In the last class, Miss Anderson, I think she explained you guys about the fragment DNA and fragment DNA analysis, like to check STR profiling, right? Right, Abu, STR profiling.
Yes,
so this STR
that's correct.
profiling,
Yeah.
STR profiling, we should do by like fragmented analysis. We do by this Sanger sequencer. So in that case, STR profiling, any one of the primer, forward or reverse primer, it will be tagged with dye like hex, wick, different dyes are there. So it will tag with that. Okay, so you do PCR and just do the purification PCR and run in this capillary gel electrophoresis. So you will have the specific target region. How you will get PCR target region in the gel? You will get like specific base pair, like 420 base pair or 350 base pair of your amplicon size. Same thing you will get capillary in the capillary electrophoresis. You will get the peak of that fragment analysis. But don't confuse about Sanger sequencing, that is nucleotide sequencing, and fragment analysis. It's the principle is little bit different. Okay, for Sanger sequencing, you will add ddNTPs, specific fluorescence, fluorophore dyes to each nucleotide. But for fragment analysis, you just label the primer. So hope you got Sanger sequencing.
Yes, yes. Now
This is the, yeah, this is the electrophoregram. So here you can see starting. Here you can see starting. You will have the very bad quality. Then you will have the good quality. Actually, this, I'm sorry that this picture is not so good. So okay, now I'll go to the NGS. Okay, so NGS, this is you have different type of NGS chemistries. Okay, this chemistry called clonal amplification, and this is called bridge amplification. First thing, any DNA sequencing, when you want to start, you will take the DNA, purified DNA. Okay, then you will do the fragmentation. Okay, this fragmentation, why we will do this fragmentation? To chop this DNA with small pieces. Okay, so you will have big chunk of DNA, so you have to chop this DNA with small pieces. That is we call fragmentation, and that fragmentation can be done by different method. It can be done by enzymatic method. It can be done by sonication. So by sound waves you break this DNA with small pieces. Okay, and you can standardize this sonication or fragmentation by adding different concentration of enzyme, or by for sonication you have to give the different duration of the time for sonication. Okay, so you small piece of large piece of DNA. So by this after fragmentation, you can check this fragmentation on the gel. So you will have it's like a strip of fragmented DNA on the gel. After fragmentation, we should do end repair and adapter ligation, right? So end repair I will explain here. So first you have to extract the sample, then you have to create DNA fragmented, that is you do by sonication and enzymatic digestion. Then we have to repair the ends of the DNA, of the breakage DNA, that you do by adding some enzyme called clinofragment or T7 polymerase to form the blunt end. Okay, you know what is meant by blunt end and cohesive end for in the DNA. So blunt end is like the two strands have the same size, end of the size, but the blunt end will have some the overhangs. Sorry, cohesive ends, you have the overhangs of the DNA. So for that you have to repair it by adding enzymes, then add the adapters. So adapters will have what will have it will have the indexing, and it will have the ligation of adapter sequence which will bind to the flow cells. Okay, you know what is meant by indexing? Anyone knows what is meant by index?
As far I know, indexing means for one samples, we are putting some sequencing that will be specific for that sample, so that we know, okay, this sample is focused.
Yes, so the indexing is like you will have, like it's like barcoding. Okay, so specific, say for example, Abu samples we index with specific barcodes. All the fragmented DNA will be barcoded by specific indexing for the Abu sample. Adil sample, for example, we will barcode with other indexing, other sequencing. So it is just sequence actually. It is 6 to 7 base pair sequence, which will ligate it to the fragmented samples. Okay, so here you will add adapters. Actually, adapters has two sequences. One is the adapter which will bind to the flow cell, and inside the adapters one more sequence is there. It will have the index, so that in later after sequencing we will demultiplex. What we call, actually here what we do is we mix up the sample after fragmentation. After adapter ligation, we mix up the sample. Like one flow cell, we will run maybe 16 sample or 20 samples in one flow cell. So we mix up the sample. So, but later after analysis NGS, we separate those samples by bioinformatically. Okay, we separate those samples and doing analysis for specific, for example, for Abu Bakr Siddiq or for Adil, we separate those, separate reads for specific for Abu and separate reads specific for Adil. Okay, so this is the end repairing and adapter ligation. Okay, then I will go with different PCR which will do for NGS. Okay, so here you can see different platform. You can see pyrosequencing, which will used by Roche and 454 sequencing. Then we have the ligation that is called pyrosequence, that is sequencing by ligation, that is solid sequencing, and ion torrent, that is from thermo sequencer, ion torrent, and Illumina reverse dye-terminated sequence. Illumina is by reverse dye-terminator is Illumina. So first three is by emulsion PCR. I'll explain next slide by what is the emulsion PCR, but reverse emitter is the cluster based sequencing. You will form the clusters. Okay, pyrosequencing output is luminescence. This is the detection of the NGS. Then solid, that is ligation, that is fluorescence, and ion torrent, pH change, and reversible dye-terminated Illumina, that is fluorescence. So in maybe in ASCP they might ask what is the output detection criteria for different NGS platform, like pyrosequencing by luminescence, okay, and sequencing by ligation by fluorescence, and ion torrent, that is by pH change. You will have pH change in the reaction, and reverse dye-terminated by fluorescence. Okay, then so what is mean by chlorine amplification by emulsion PCR? So emulsion PCR, it's by, you will have in the reaction mixture, you have, okay, first you do fragmentation. The third step you will do library preparation. In library preparation you will do the different amplification. Here we can see clonal amplification. So you will have in the for example in the PCR tubes you will have small beads. Okay, we call like it can be magnetic beads, something. In that magnetic beads you will have it specific probes which will ligate the fragmented DNA. Okay, so this is the beads, right? So in each beads, okay, there is amplification is going on inside the chip. For example, here you can see, so each beads will have different probes or primers, okay, which will ligate the specific targeted sequence. Okay, then but what will happen? So each emulsion PCR you will have different multiplex happening. Like same thing like what when you do general PCR, you will have specific targeted PCR amplification. But here in emulsion PCR, different primers and different probes will amplify the specific region of interest. Okay. Then this is sequencing by ligation. This is solid sequencer. Here what they use two probes. One is primer 1 and primer 2. Here you will have the ligase based sequencing. But presently you should not bother about this. This sequencer is not, it is outdated. Nobody is using this technology, sequencing by solid sequencing. Then the third one is, this is pyrosequencing. So pyrosequencing, what here we use different enzyme. One is apyrase, and one is here the sulfurase. Okay, when when there is incorporation of DNT, when there is incorporation of DNTPs, there will be formation of ATPs. So here you can see when there is incorporation of DNTPs into the, when polymerase add DNTPs, there is a generating ADPs or AMP and pyrophosphate. Okay, so this pyrophosphate, what happens is pyrophosphate in presence of sulfurylase with TPI, it will form ATP. So this ATP with the having with the presence of luciferase enzyme, it converts luciferin to oxyluciferin. So this will present the light. Okay, then you will have the pyrogram. You will have the pyrogram, and this question, because I have taken the SMB ASCP. This question they have asked how to read the pyrogram. Okay, so in the pyrogram, for example, if it is AA, you will have one peak, bigger peak. If it is different nucleotide, you will have single peak in the pyrogram. So they will ask different in the exam they will ask different pyrogram and identify which sequence it is. Okay. Now this proton based detection, that is H plus ions, that is this is by ion proton, that is from Thermo Fisher. So what happened when there is incorporation of nucleotides, DNTPs, there will be H plus ions, there will be H plus ion generated or synthesized, generated in the flows in the reaction. Okay, so each ions, when it incorporated, the camera will read which ions has been detected. For example, here I'll show you in this how this ion sensitive field effect transistor happen. Actually this is present in the chip actually. Okay, so what happens when there is H plus ion generated, there will be a transit of pH change. So here you can see when they add the different nucleotide, so there will be a H plus ion generated. So based on this H plus ions, okay, based on the H plus ion, the pH is detected. But these questions I have asked to the companies, how the machines knows that, so each nucleotide will have the H plus ions, right? But how this machine knows how the sequencing happening here, means which nucleotide is changing. But actually speaking, the how much number of nucleotide has been present there. So it detect by pH as well as how many nucleotide it get pumped. Okay, this is actually ion sensitive ion tolerant base. It detect by pH change. And this is the last, this is by bridge amplification. So bridge amplification by which NGS, anyone? Bridge amplification, anyone knows bridge amplification?
Illumina?
Bridge amplification, bridge clonal amplification by bridge PCR.
Yeah, this is by Illumina.
Yeah, this is by Illumina. Yes, correct. This is by Illumina. So when you do the library preparation, that is by adding the adapters ligation, so you will add this library into the solid surface we call flow cells. In flow cells there is a cluster generation. So here you can see after adding the adapters with the insert, you will have cluster generation. Okay, so, and this flow cells, it has probe which will identical to that adapters. So you will have here PCR based bridge amplification, like this bridge. Okay, so you will have, and this is by synthesis, we call synthesis by SSS, synthesis. I forgot the specific term. I'll get back to you later. Cluster, this is actually when we add the library into the flow cell. Actually this is a flow cell. You will have the cluster of DNA molecule generated, we call colony or colony. Okay, so you will have the cluster of sequencing. So there's millions of copies of specific PCR is generated in this flow cell. Okay, and this is so when we call cyclic reversible terminator. So what will happen here when there is different fluorescence labeled DNTPs has added. So each DNTP is captured by the camera, CCD camera. Okay, so this is by cyclic reversible termination. So this is by Illumina. Fine. Now in market, you will have different sequencer, Illumina HiSeq, Illumina MiSeq. So Illumina HiSeq to sequence, high amount of DNA, high amount of data to be generated by Illumina HiSeq. Illumina MiSeq can be generated by very less amount of data. Okay, and you have here ion proton, that is by Thermo Fisher, and Roche 54 and ABI Solid 4. This is actually presently nobody is outdated, is not buying, and this is called PacBio. I have not added any slides for PacBio. This is actually long read sequencing. Okay, and just remember that in sequencing there is short read sequencing and there is long read sequencing. So Illumina, Thermo Fisher, Pyro sequencing, all these are short read sequencing. PacBio, it's long read sequencing, like it can run, you can sequence 10 kb at a time by PacBio. But Illumina, that is, we have to sequence 150 base pair at a time. So you will have 150 base pair short reads. Okay. So which is the best sequencer? So it's all depend on the project you are working on, or it all depend on the clinical diagnostics companies or clinical diagnostic lab, what they are targeting, what which application they are using. So based on that, they have to choose the which sequencer they have to purchase. So here you can see different read length, accuracy. Okay, so you can see here at a time you can at least 2900 base pair you can sequence by passive bio. Ion torrent 200 base pair, pyro sequencing 700 base pair. Okay, and sequencing by synthesis, that is 50 to 250 base pair. So different, and Sanger sequencing you can sequence up to 4 to 900 base pair. So you can see the cost, you can see the reads per run, time per run. So different technologies has different read run, accuracy, and the cost and its disadvantages. So here this slide shows the what you can do by using different technology like whole genome sequencing, de novo, RNA-seq, chip sequencing, amplicon, methylation. I'm not going. I'll go detail about on exome sequencing in my next slides. Okay. This I'm not going. Okay. Now in exam ASCP they ask FASTQ files. Okay, how you store the files? See, FASTQ files are those files when it's coming out of from the machine. Okay, in FASTQ files you will have this something scored called we call Fred quality scores, and in NGS in ASCP they have many times they will ask what is which file used for what. Okay, so FASTQ files used for actually it will use for storing purpose, and this is the starting file which is coming out of the NGS machine, and what is called Fred's quality scores. So here we can say Fred quality score Q20, Q30. If it is Q20, it is the error rate. Okay, 1 in 1000 base pair it has a error, and the base calling accuracy is 99.9%. If it is Q20, 1 in 100%. So 1 in 100 base pair you will have some error. Okay, some error in the sequencing. So the accuracy is 99%. So this is the Fred quality score. So everyone knows about the Fred quality score, right?
Yeah, to understand like how good is the sequencing.
Yeah, so to know what is the error rate, so it has, see, NGS has more error than Sanger sequencing. Sanger sequencing has less error, but NGS has more error. Okay, so based on by looking the quality Q30 or Q20 in the, you can tell that this is the quality of the sequence. Okay, so each sequence, it has each nucleotide, it has a quality score. So they will take average of all the sequence, and it will give the quality, Fred quality score we call. And what is the coverage? Anyone knows what is the coverage?
Depth of reads.
Yeah, its depth of number of reads has been run in the specific
How many times?
sequence. If, for example, you can see yeah, how many times it has run, how many times the reads has been run in the specific region. Okay, so this is the coverage. This is actually screenshot of IGV. Okay, and this is the, it has been mapped on the read mapped on a reference genome. Okay, so you will have, we will say 30x in the reads, you will say 30x means 30 times the reads is, has run in the NGS. I'll show you next time how you so for example this. So what happens when your data is out from the FASTQ, you will run, you will actually you assemble on the reference genome. So for example, here you have reference genome, this is a reference genome, green color. Okay, and this is the different size of the reads. Okay, for example, coverages means of reads underlying the consensus, how many number of times it has run the sequencing. Okay, now here you can see this G at specific target, it is 6x, means 6 times it has run, so we call 6x coverage, so it is 100% identity. Okay, if it is any one miss here, for example, here it has come A to C. Okay, so this is not 100% identity, here it is 99.9% or 99% identity. Okay, so here you can see, so here they say 80% identity, so it is 5x, 1 2 3 4, so 5 times it has run, and it is 80% identity. So this might be a mutation or any variant here. So this is 2x coverage, 50% identity, and this is 1x. Okay, so when you do assembly, so actually after having we use different tools for mapping the genes, mapping the sequence in the reference genome, like SAM tools, then we have BAM file, BAM tools, then at the end we call it VCF file. I will tell you what is this. So here just it is the aligning the sequence on the reference genome. Okay. Can you hear?
Yes, we can hear you. Sometimes it is broken, but we're gonna adjust. I think we can adjust. It's okay.
Okay. Now I'll go. I have some other slides also, but with time permit, I can explain.
Yeah, sure. Take your time, no worries.
1 minute.
It's okay. Like, yeah, like I think
Okay. Okay.
we are okay.
Yeah. What we can do is then see. I'll explain you some of the application of NGS. Okay. I'll tell you some of the application of NGS. So the so here you can see whole sequencing. So what actually whole exome sequencing is, what we are sequencing all the exons of the human genome. It is called whole exome because, as you see, why we are sequencing whole exome, because as we know, we have around 20,000 genes. Okay, and we have around 3 billion base pair in our genome. We have around 3 billion base pair. This question they will ask, what is the how much billion base pair, or how much base pair we have in our genome. So we have around 3 billion base pair. In that 3 billion base pair, only 1.5%, okay, maybe 30,000 3 million base pair is our exons, that is coding region, that is around 180,000 exons are there in our exome. Okay, fine. And 85% of this 85% of the known causing mutation in our human is present located on exon. That's why we do whole exome sequencing. And whole exome sequencing mainly we use for all these Mendelian diseases. Mendelian diseases, like for example, you can see like Fragile X, thalassemia. This is all basic diseases like sickle cell anemia. But there are lots of diseases. There are around 7,000 Mendelian diseases. Okay, and I did my PhD on my PhD on whole exome sequencing analysis. So we have around 7,000 Mendelian diseases, out of which to identify the genes and the mutation is very difficult by Sanger sequencing. So that's why we use whole exome sequencing to identify the mutations. Okay, and as all you know, me, like every human beings, we are actually, in terms of genetically, we are similar. We are like 99.9% we are similar to each other. The 0.1%, it will give us our, our color of the skin, color of the eye, color of the, hair. It's all actually, we are 19.9% we are similar, and 0.1% is our different, color, height, race, everything. Okay, and the disease also in that 0.1%, if any change in the coding region is going to have the disease. Okay, then next, and this mutation is very conserved in across species of protein. Okay, that's why we do whole exome sequencing, and whole exome sequencing is cheaper than whole genome sequencing. Is 1/6 cost of whole genome sequencing. Whole exome sequencing is, and this slides tell us whole exome sequencing. As I said you, the whole genome is, the size is 3.3 GB, that is 3 billion base pair, and whole exome is 50 MB, that is around 1% or 1.5%. Okay, whole exome sequencing. In that we are sorry, exome is 1%. In that we take only exonic region. So as you know, in the exon we have exon and intron, right? So we sequence in whole exome, we sequence only exonic region of each genes. Okay, so all the 20,000 genes is been sequencing by whole exome. That's why we say multiplex sequencing or next gen sequencing. At a time we sequence all the genes region in our exome. Okay. Now this slide is just to show traditional sequencing and next gen sequencing. In PCR based or Sanger sequencing, we take single genes, like maybe BRCA1. We can sequence BRCA1 by Sanger if it is very small gene. Okay, with small gene we can sequence by Sanger sequencing. But if you want to sequence many genes like target region or exome sequencing, you should take NGS. Okay, for example, some panels, they are called panels, like NGS panel for example, retinitis pigmentosa, or for example any neurological diseases. Okay, there are specific panels are there which can cover all the genes which are associated for those diseases. Okay, so in capillary sequencing you will get like this Sanger sequencing, like peaks. Okay, but here you will get, in NGS you will get like this read depth. In this read depth you can see by viewing the IGV. Now whom we have to offer the whole exome sequencing? In basically it offers for genetic basis, like I said, Mendelian diseases. Like Mendelian diseases are those having the one gene one effect, one gene one disease. Okay, and with significant genetic heterogeneity. Genetic heterogeneity means one disease has different genes. Okay, for example, Joubert syndrome, it has many genes which cause the Joubert syndrome. Or overlapping clinical features or atypical clinical presentation and complex phenotype with multiple differential diagnoses. So clinicians or clinical geneticists, they are confused which disease it is, because as you know, there are 7,000 diseases are there, and maybe more than 8,000 genes are there to to identify. So what we do, that's why we will directly go for whole exome sequencing or unsolved genetic testing such as normal karyotype, microarray, and all the techniques like MLPA, single genes, clinical exome, everything. While after doing this, there is no answers for the families of the of the child. Okay, and heterogeneous phenotype like intellectual disability, development delay, multiple congenital anomalies, and different diseases like cardiomyopathy, epilepsy, undiagnosed metabolic disorders. So all, if you in your America, if you see NIH or National Human Genome Institute, NHGRH, I think, all NIH projects for this pediatric diseases, they are finding a new genes for this Mendelian diseases. I'll give one example. Now, this is actually Joubert syndrome. Okay, it has different symptoms. These are all symptoms. Okay, Joubert syndrome has congenital liver fibrosis. It has Joubert syndrome plus liver, and it affects kidney. So it has different symptoms. Okay, these are all the symptoms. But the now, and there's a different types of Joubert syndrome. So there are different genes. So you can see there are 9 to 10 genes. So it's very difficult to do single sequencing. So directly what we can do whole exome sequencing. Which gene it has. For example, this is one case. While I was doing whole exome analysis sequencing, I will show you. So we got a mutation in AHI1. Okay. So there is a stop codon mutation. So any whole exome sequencing, you will find like this. Okay, you will have gene, and this is the transcript ID. Transcript ID, you can, this is NST, means ensemble ID. These are two types of IDs in transcript. This also they will ask in ASCP, especially in SMB. What is called NMID? What it depict? It's depict actually cDNA. Okay, so you can depict by NMID, or you can depict by ensemble ID. And this is the chromosome coordinates, and this is the variant change of the. So you know the nomenclature, how to nomenclate this variant. For example, this variant is 12962G. So this is the C position at this gene. Okay, and this is the protein change and zygosity. So these are two types. Zygosity is homozygous and heterozygous. Homozygous means the change in both the chromosomes or both the DNA. It came from father and mother. And this is the homing phenotype, inheritance, and this is the clinical significance. So we classify the clinical significance by different 5 nomenclature, like pathogenic, likely pathogenic. Okay, VUS, that is called variant of uncertain significance, benign, and benign, likely benign. So these are this is the example of the report of the whole exome sequencing. So all this classification analysis we do by ACMG guidelines, American College of Medical Genetics. Okay. So briefly, I will show you how we do whole exome sequencing analysis. So anybody has any doubts?
It is clear to me.
Hello?
If anybody has any question or
Because now because why I took whole exome sequencing, this is the now market in the America or anywhere, USA, Europe, or anywhere. Whole exome sequencing is now. Hello?
sorry,
The
anybody
whole exome sequencing.
anybody said anything? If not, I can share one of the things. Like in New York Genome Center, they normally do, I think, this whole exome sequencing for the prenatal. Like the woman will be stay pregnant or not. They try to define that, like to find out any like life-threatening disease or something like that. I think they try to do that. Yeah. But my question is like, how much is the cost? Like, is it reducing day by day or something?
Yeah, that is That is it.
Hello.
Yeah, it's reducing. For example, see, now in the market it is maybe $400 to $350 for one sample. For one sample, maybe $350 to $400.
That's why I think it is getting
Hello,
more and
can
more.
you hear?
Yes, we can hear you.
Hello.
Yes, we can hear you. Uh,
Yeah, yeah, it's all depend. It's all depend on company to company. It's all depend on the qualities. Okay. If what they do is what they do is if they reduce the price of that test, for example, whole exome sequencing, but the quality will decrease. Okay, why the quality will decrease? They will add more number of samples in that chip. Okay, if they add more number of samples in that chip, in that flow cell, okay, so but quality will decrease because the coverage will decrease there. Because at least for whole exome sequencing, you should have 80 to 120x coverage depth across whole exome. You can hear me?
yes, we can hear you.
Yeah, so at least we should have 80 to 120x for whole exome sequencing, at least to find to show that, or if it is like 30x or 20x, we will miss out some variants, we'll miss out some sequencing. Okay,
That's right.
that's I will show you some annotation file also. One second. 1 minute. I'll show you how to do analysis. Because whole exome sequencing, it's now analysis. You can work after your master degree. If you had done master in genomics or genetics, human genetics, you can work as a variant scientist or a variant genome analyst. But I work after my PhD. I work as first job is my senior genome analyst. So we used to work. There's a company called MedGenome. It's there in America also. It is there in India. So I work in analysis of whole exome sequencing. Every day we used to analyze 2 to 3 samples per day. Even now also I do some work on whole exome sequencing by checking the annotated files. Okay, so how we do is whole exome sequencing. I will just show you. So see, when we have whole exome sequencing, you will have lots of variants. You will have maybe 100,000 variants in your annotated file. So you know what is mean by annotated file, right? Hello?
Annotation means like we are adding some of the like gene name or something, like we are annotating one of the sequence, right?
Yeah, it's like after sequencing you will have just sequence, but you annotate different, after doing reference genome, assembled in reference genome, then by bioinformatically you can annotate genes name, exon, where this exons is, and which what is the change in that region, whether it is a stop codon, frame shift mutation. Okay, everything. I will show you one of the example. I'll share my Excel file, one of the Excel file. I'll show you. 1 minute.
Okay, no problem. Take your time.
Till then, anyone has any doubts?
Um, I have
No,
a
doctor.
question.
Yeah, Adil can ask, no problem. Adil has doubt or Abu has doubt?
No, Abu Bakr.
Yeah, I
Okay.
have one question.
Yeah.
Sorry. So my question was like you said Sanger sequencing cannot find Mendelian disease. Is it
No, it
because
can
of
find. No. It can find. It can find Mendelian disease, but you can't see if you know the disease. For example, if you know the disease like upper syndrome. Upper syndrome has only one mutation, hotspot mutation.
Hmm.
So you can sequence only hotspot mutations by Sanger sequencing. But if you don't know which gene it is, or you don't know where the mutation is, then you have to go by NGS. Okay.
Got that?
Okay. Okay. And the other example is like if your PhD project is like you have project, maybe you started PhD and and you are working with specific disease, like for example I work on MPS 4A, it is called Morquio syndrome, and it contains only one gene called GLNS gene, galactosamine acetylase. Okay, so GLNS gene. So that gene has only 14 exons. So in my PhD, I design a primer for only coding region of that gene. Okay, okay.
Hmm.
I just design a primer and do Sanger sequencing and identify the mutation, because we know the other, because I work with the clinicians, with
Mhm.
the medical geneticists. Okay, so the medical geneticists know this is the disease by phenotypically, by by clinically, they diagnose this is a disease, but they don't know where is the mutation. So we do Sanger sequencing to sequence all the region in sequence. If you have a less number of exons, you can do by Sanger sequencing. Okay, but like 8 to 10 years before that time, whole exome sequencing was very costly, and nobody were using it very lessly. They were using more on Sanger sequencer. So we use that Sanger sequencer to sequence 14 exon in that gene, or maybe 20 exon in that gene. But it is laborsome, it is cumbersome. You need lots of time to sequence Sanger sequencing. As you know, to sequence whole genome, it took maybe 13 years to 14 years. There are two, not one machine in Sanger sequence. There are lots of machine they use in NIH project. Okay, so you can do, but you can't do more sample, and it is like it's not a very fast Sanger sequencing, takes months to do. It is one or two mutation you want to check, you can do by Sanger sequencing, no problem. Okay, and the one more thing is good thing is additional good thing is Sanger sequencing is to confirm. For example, you find the mutation in a child. Now you want to confirm whether that mutation is segregated from the family, from the father and mother. That time you should do Sanger sequencing. Okay,
Okay, that sounds
you
good.
got my point?
Yeah, yeah. Now
Yeah,
it's
because
make more
yeah,
clear.
because it is, if it is autosomal recessive disease. Okay, if autosomal recessive disease like thalassemia, okay, sickle cell anemia, sickle cell anemia, sickle cell anemia, it is present in HBB gene. So you sequence specific that exon, you find the mutation in child, then you have to sequence now father and mother. Okay, that time you do Sanger sequence. You don't do whole exome in father and mother, but it will cost more for them. It's all about what is the strategy, which technology to use. So when you become after PhD, and when you started in diagnostic lab and you work it, so it all depends what is your strategy to identify the mutations, which techniques you want to use. For example, you can't use MLPA to identify a mutation. Okay, so MLPA will be used to identify the copy number variation, CNVs, copy number variation, for that is MLPA, right? And microarray, it will be you can use to check the deletion and duplication that they use for not all the disease, they use for when there is abortions, miscarriages, you use microarray. You got my point. Previously they were using karyotype, now they are using microarray.
So all
Okay.
sort of de novo mutations, yeah, the diseases associated with de novo mutations, we can use NGS.
Yeah, if the mutation is de novo, you can use NGS. Yes, yes, de novo mutation first time it is present in the child, you can use NGS. And for example, epilepsy, mostly the epilepsy cases and autism cases, you will have the de novo. mutations. Okay. Now I will show you one of the. It's just it will take difficult. You will have annotate file. Just okay. First I will explain you how we do filtering out the common variant. See, whole exome sequencing. What we do is we filter out the common variant. Okay, common variant means it's called polymorphism. It's like single nucleotide polymorphism. Okay, it's common in normal population, but we have to filter out those common variant and take only rare variant. Okay, first we will have, after annotation, we will have Excel file. I will share this Excel file. I'll share this Excel file. Share content. You can see this Excel file.
No, I think you can stop share. Now we can see. Yes, we can see.
Yeah. See, in annotation file, actually this is actually couple cases. This is like just Excel, but you will get very difficult to read. See, you will have here chromosome number. This is genomic coordinates. Okay, you will have this reference and ALT. Reference means reference genome, and ALT means change in the sequence. Okay, you will have here read depth. See, here you have 73 read depth, 78. Okay, actually this is actually couple case. Actually the couple has severe abortions in the child. They have miscarriages, they have neonatal death, the children. So it came for carrier analysis. Okay, so you will have couple like father and mother, husband and wife, sequencing. So we do combined sequencing. Okay, so this is the, you will get like the zygosity, heterozygous, homozygous. So, and you will do filters mostly on the, put filters on the wait, I'll show you on this filters. Genomat is the whole genome database. Okay, so there's no time to explain all the columns. So actually this is the annotated file. So when you do after NGS final file, you will get the annotated file like Excel. But most of the companies, and they have now software to analyze. So what they do, they upload this file into the software. So you can put the filters, you can put the genes, you can put the different ontology, different phenotypes in this filter. So now what I will with my slides, how to put the filters. So you have to put the filters, you have to remove the depth more. Basic filters, you take the depth 3x, more you take the variant. So less than 3x, you just ignore the variant. And this is a map, minor allele frequency. Minor frequency means, so we have to take the rare variant. So less than more than 1%, we rule out the variant. Okay, more than 1% of the genome, we rule out the variant. From that exact, I said exact is the genome database. Exact, genome, at 1000 genome projects, internal database which from the companies or any institute. And you take only transcript type, like protein coding, that is missense mutation, termination, frame shift, everything. And this is the variant position. It should be present in exonic region, deep reported intronic, or you can take UTR region, but most of the mutation will be present in the exonic region. So you remove all the common variant and find the significant variant. Then another thing is you should consider when you do analysis is in silico evaluation. This is all bioinformatics analysis. That is how the mutation is, it has effect on proteins, those things we take, and whether this mutation is published in literature, in some papers. And we check segregation information. Segregation information means whether that mutation, after reporting that mutation, we tell to do, we inform the clinicians to do the their father and mother Sanger sequencing. And we check the population database, that is minor frequency. For this, you have to read this paper actually, ACMG guidelines. If you read this ACMG guidelines, you will know this is it's available in Google. You can just search ACMG guideline for variant analysis and interpretation. But this one, by reading this, you will get the ideas how to do analysis, what is the pipeline, what to follow the analysis, interpretation. But to get the the to get the idea, you should have the experience and learn how to do filtering out the variant and to do interpretation and reporting of whole exome sequencing data. The other is application of NGS is NIPS, non-invasive prenatal screening. So this non-invasive prenatal screening is a safe method by just taking the blood of the pregnant woman. Okay, this might be asked in ASCP about this NIPS. So as you know, when the woman is pregnant, okay, after 10 weeks of gestation, okay, we can take the blood of the pregnant woman and check the any abnormalities of what, of trisomies, or like structure abnormalities like trisomy 13, 18 and X and Y. Okay, this 5 chromosome abnormality we can check by taking the human maternal blood. Okay, so from that we can extract the, as you know, there is a cell-free fetal DNA in the maternal circulation. Okay, so you got this point, no?
Yes,
we can take
yes.
we can take cell-free DNA. This is called, now this is using very rapidly. Many centers, many hospitals are using this at the age of at the 10 week of gestation. They just take the blood of the mother of 10 ml blood and extract the DNA, and just they do the NGS sequencing of use. There'll be a mixed up DNA there actually, cell-free DNA as well as of the child and of the maternal DNA. It will mix up, but they do bioinformatically, they separate the cell-free DNA and maternal DNA reads. Okay, so to whom to offer this NIPS? Actually, they do for maternal age. So most of this 21, Edwards syndrome, Patau syndrome, and our trisomy 21. That is what is it? Trisomy 21 is which one? Down syndrome. Okay, Down syndrome. This will occurs actually more than 35 years when the pregnant woman is risk of having more than 35 years of age, and positive serum screening. So they do by doing biochemically, they detect positive serum screening. Okay, and abnormal ultrasound reports. If there is any in ultrasound, if they detect some markers for this trisomies, we can do NIPS, and previous history of miscarriages or family history of this trisomies. Okay, and this is a just template of the report, how we report like low risk, high risk. If there is if any reads on trisomy 21, we read as high risk, and or else we just read as low risk. And we can do NGS, we can do PGT. PGT is pre-implantation genetic testing, pre-implantation genetic screening, or preliminary genetic diagnosis. This, as you know, this is also one of the clinical. We can do the NGS by checking the embryos for the IVF centers. In IVF centers, when before implanting any embryos, okay, they can check any abnormalities in the embryos. We can check by NGS. That we do by NGS, and check any CNVs, copy number variation, by you can do the PGS NGS sequencing. So to whom we offer this pre-implanted genetic testing? Okay, every pregnancy has the risk of miscarriages in the first trimester, or the risk is further increase in IVF pregnancies. Means being pregnant, she goes to the IVF centers to have to do the IVF, and there is anything like increased risk of IVF pregnancy. There's a lots of rejection in IVF pregnancy, and there's lots of miscarriages in majority due to aneuploidies in the fetus. I mean, most of this rejection and any abnormalities or any abortions happens or any miscarriages happens is due to the aneuploidies in the fetuses or in the embryos. So what we do is we do by two method. We can do by PGS, that is to identify on the right side, to identify the chromosome abnormalities. We can do PGT, that is to know the Mendelian genetic diseases. Means PGT means pre-implanted genetic testing or pre-implanted genetic diagnosis for Mendelian diseases. For example, if someone has thalassemia in the family, a child has thalassemia, if they want to do you can do two things here. Okay, you can do prenatal diagnosis, that is of prenatal diagnosis means after having a baby inside the womb, we can take chorionic villus sampling, or we can do amniotic fluid and test the specific mutation by Sanger sequencing if you know the mutation. Or else is some populations, like for example now in Saudi Arabia or Middle East, there is a ethical issues for abortions, or to terminate the baby because of religious things. So what they do is they go for IVF, and what we do is we take the embryos and we check the specific mutation in specific all the embryos, whether the mutation is there or not. So if there is no mutation, that embryo will be implanted. Okay. So how we do is we do it at 3 days and 5 days. Okay. On third day we can do, or 5 day we can do. If you do on third day, we have to give the report on 5th day, and they have to implant. Or some countries, in for example America, what they do is they do on 5th day, because they don't they freeze the embryos, and the next their menstruation period they implant the embryo. Okay, so what we do is they do biopsies from this third day or 5th day. So they take the small small cells, 2 to 3 cells of the embryo, and send to lab to do NGS. So they send by, like, they have this kit to transport. So what happens when we do NGS for this embryos, we can detect trisomies, monosomies. Here you can see in the first, on the left side, this is a normal this is normal cyto, we called what do you call, we call because this one not electrophotogram, this is called cytogram. We can check any CNS. Here you can see there is no loss and gain of the region, but here you can see in monosomy, here at 7 region there is a deletion here, and this is a duplication here. So this is the monosomy of 7 and trisomy of 14. And here you can see mosaic of chromosome 10 and 18. If there is a mosaic, you will have the it's made between mosaicism means some of the some of the cells in the embryo it's has deleted, and some of the cells is normal. So you will have like this ratio here, and this is the loss of DNA in the right side down. This is the loss of DNA, so you will not get good data. And how you report the PGS like this. For example, if you get some 6 embryos to the lab, we report as euploid. Euploid means normal embryo with XX. XX is a female and XY is a male. You know that. If it is aneuploid, we report like aneuploid XY plus 1 plus 2. So there is a trisomy of 1 and 2. Okay, and we recommend not to transfer. If it is good embryos, we report as euploid and recombinant. So what they do is the embryologist they inform the OB-gynecologist to transfer any of the 3 embryos. Okay, it's all sometimes this is like to do sex balancing in the family. If the family has more females, okay, so they can choose male embryos and to have a male baby. So this is all my presentation. I just want to wrap up because this is actually maybe too much for you guys. Any questions?
Thank you so much, Dr. Mohit. I have a lot of questions, but I think it's a like
You can ask, no problem. If If anybody wants to leave, you can leave, or if they want to stay, they can stay.
Okay. If anybody has any question, please unmute yourself and ask. Or you can write in the chat also. It's okay. I think when you were talking, one of the members named Sanap shared that they performed Sanger sequencing for beta globin gene. So I think when you asked like anybody did any Sanger
HBB gene.
sequencing or not, yeah.
Yes.
So that is one update. So that's good. I think we can. So my question were like you showed in the Excel that there has like both father and mother.
Yeah. So yes, that is actually couple. That is actually couple samples. So what happens is when couple has lots of abortions, miscarriage, and by ultrasound they by ultrasound of the before miscarriage they found something anomalies in the child or something very deleterious, very very harmful, maybe have phenotypes in the child. So what they do is they send couple samples. So what we do is we analyze the genes. So we have a set of genes for abortions, maybe any aborted, very deleterious genes which have the which might the mutation in that gene. So what we take is we take the common mutation in that gene. Okay, the common. So it can be two types of mutations. Okay, there is a homozygous mutation in that gene, or we call compound heterozygous. Compound heterozygous means two mutation in that gene. For example, in if you take HBB gene, one mutation can be present in exon 1 and second mutation can be present in exon 2. Okay. Okay, in NGS you can check whether that mutation is cis or trans. You know what is mean by cis or trans?
Cis is in similar similar.
No, cis means the mutation present in the same read, or the same mutation present in the same strand, okay, or the same chromosomes, okay. Because when we sequence it we can detect, we can do by just sequencing, maybe if the mutation is nearby, okay. Because when we do Illumina sequencing, we sequence 150 base pair, okay. If that mutation present is nearby, maybe 10 nucleotide base pair apart, or 15 nucleotide, so we can check by cis whether it is cis or trans. Cis means if it is present in the same read, so that it is present in same, like haplotype means same sequencing read. Trans means two reads, different reads. One is present in other read, and one, because in Illumina we do bidirectional sequencing. Sorry, we do pair read sequence, not bidirectional. We do pair read sequencing, read 1 and read 2. Okay, so in the if you see IGV, if you have chance to see IGV, it's there in some of the books. IGV, that is Genomic Viewer, International Genomic Viewer. There's some books recently you share, right, about the clinical molecular genetics for the some question and answers. In that book there is IGV, and that book actually is good, which you shared is good, but that book it's very more depth on genetics. Okay, if you want to learn that books genetics, that is a good book. But in ASCP, as I know, they will not ask only genetics, they'll ask
Everything.
microbiology, everything. So you should have what I feel suggest everybody is like, for SMB they should read everything and with 100% sure, then only they can take the exam and pass the exam. Because as I know, as I told you before, that they will focus more on what is your weakness. The computer see first 20-25 question, it will see what is your weakness, and they'll ask more on that topic. Because I'm good in NGS and genetics, because I have experience in more on genetics. So if I answer well in genetics, they will not ask more on genetics, they'll ask other topics.
Yeah, that's right. That's right. That's why it's a like CAT,
Okay,
computer adaptive.
so
Yeah.
about your couple, you got my answer about couple analysis.
Yes, yes. It answered my questions. Anybody else has any question?
So whole exome sequencing, actually what we do is we have set of genes for specific panels. For example, by checking the looking the symptoms of that the child has. Okay, so what we do is we have set of genes for maybe retinitis pigmentosa. We have set of genes for neurological diseases. We have certain genes for kidney diseases. We have specific all genes, maybe 300 genes panel. Okay, we have our gene panels in our database, or we have the set of genes in my laptop. So what we do is I will put the filters. So I remove the common variant. Okay, and out of 100,000 variant, when I sequence whole exome sequencing, the person will have 100,000 variant or 200,000 variants. So I filter out the variant and come about maybe 10,000 variant at the end. Okay, after 10,000 variant, then I put the filter for the genes. That is called, we call genotype phenotype correlation. I put the filter for 300 genes, so it will select only those 300 gene in that data. Okay, after that what we do is at the end we got maybe 10 to 15 genes. Then we will do the variant interpretations, whether that variant is matching with the phenotype. We match that variant present in that database, that data with the omim phenotype. This is called, you know about omim, right? On land Mendelian inheritance of man. So we, yeah. So out of from that, from the omim database with the phenotype of that child, what I am analyzing the child, we match that phenotype to that omim phenotype, whether it's matching perfectly, whether it has a partial matching. So we do variant interpretation. At the final call, we report that variant in the report.
Got that.
In America, who signed this whole exome sequence report are the fellow of American College of Medical Genetics. That's called fellowship. And they are the directors of the lab, or they are the fellowship. If you visited New York Genomics, no. So there's one Indian guy there. What is his name?
Her name is Dr.
Wait, I'll show you.
I just did the meeting with her. Jaban Putra, Dr. Jaban Putra.
Yeah.
Yes. Something like that. Yeah. So yeah, also like she is a professor of Columbia University, and she is the director of that section, like molecular testing section. So I think the last presentation, last thing that you just showed, this is normally done over there. They try to
Yeah.
check the prenatal.
Yes.
I have another question. You said last 15 base pair and first few base pair are not good.
Yeah, in the Sanger sequence, in the Sanger sequencing, so in the Sanger sequence, first few base pair, 15-20 base pair in the Sanger sequence will be not good. Okay,
Mhm.
first 15 base pair only, no, not last. First
Okay.
15 base pair it will not, because what will happen in Sanger sequencing, starting sequences will be not good. When it run in the capillary, the first 15 base pair it will not take properly. Okay, the after 15 to 20 base pair, the quality will be good. So when you design a primer, you should take your interest of region, or maybe exon you want to sequence. It should be in the middle of the when you design the amplicon, when you design the primer.
Got that. Got that. I think somebody shared in the chat that they did not use the NGS. They will use soon, and they will get the training. And yes, sure, you can learn, and also like you can ask questions in the group or in the WhatsApp. If you have WhatsApp, like you can join the WhatsApp and you can directly.
See, my thing NGS, when you work in the lab, see, there are two things. Some people work in the lab, but they know the technology of the NGS in the lab. But mainly, and you become the technologist, and you do daily every day. Okay, do daily that library preparation and running the NGS. Okay, that you do daily. But good thing is, but you should as a scientist, or when you do PhD, you will learn and you know how to analyze the data. That is main important in the future now. Main future will be NGS analysis and uh, analysis of big data. Okay, so there is now AI based artificial intelligence or machine learning based analysis. Okay, so what I feel in the future, inshallah, in the future, or when you want to do some genetics or genomics,
Mhm.
you should learn this analysis part and
Mhm.
interpretation part.
Mhm.
Okay, and you can take the course like MSc master in bioinformatics. It's very good course in US. If you they do get in MSc in bioinformatics, and you'll get good job in hospitals and all for data analysis.
That's correct. Like there is only one room of the lab. They do the sequencing thing, but their bioinformatics teams it has the whole floor or like one or two floor. I just checked, and it is very interesting because like analysis is more important. You are correct, and there has like different courses like
Yes.
computational biology or bioinformatics.
Yeah. Yes. Yes.
That's kind
That
of
is actually a good thing. That you can put your 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 mind and you enjoy different cases, and you should have very good background of genetics for analyzing the data. But when you if you're interested in the wet lab is like daily routine work. You will do same thing every day. That now robots are there, now machines are there to do to do robotic work. but
So is there
but
is
analysis, yeah.
sir, is there any platform for online platform available for data analysis which we can
No, they are
learn?
they are platform, but specific course are there. But you will learn data analysis by practice. See,
Uh,
even by bioinformatics, yeah, bioinformatics, you will learn, maybe you are learning maybe what do you call it, Python or Perl. Okay, unless we don't know where to apply this Python and Perl, and analyze the data, analyze the live data or something project data, then we can't learn nothing. There are lots of courses out there online.
that's right. Like I think Coursera, EDX, they give the
Yeah, EDS,
free
yeah, yeah,
courses.
there are
And
many things
also,
out there.
yeah, also like I think regarding learning R or Linux or Python, as you
Yeah,
said, or Perl,
yeah. You need
this
you
kind of
need you need you need you need Linux background for to run all these tools,
Mhm.
GATK, for exome. See, for exome analysis. See, there's two types of analysis. One is from downstream and upstream. Downstream from FASTQ to annotate file, that will do by bioinformatics guy. Okay, from FASTQ to annotate will do by bioinformatics. And
Mhm.
that bioinformatics, if you create the pipeline, there is a pipeline, ingest pipeline we call. If you create that in any hospital or any companies, so it's now everything will be automated. Just you have to upload the data, FASTify data, then the pipeline will do automatically, and it will give you annotated file. The next will come analysis of annotated file based on the phenotype of the patient, based on the many things are there. And even we have work on somatic mutation analysis. That is very difficult than germline analysis. So there are two types of analysis. Germline is, what is meant by germline, right? It is like
yeah.
present in, yeah, sex chromosomes. It's coming from the father and mother. And the somatic is acquired mutation, most occur in cancers, right? Like lung cancer, or might be colon cancer. Because colon cancer is more mainly on germline mutation. Mainly it occurs in lung cancer and all. Okay, sometimes then breast cancer also occurs, the acquired mutation. So we can do the somatic mutation gene sequencing or NGS by taking the tissues, FFPE tissues, fix FFPE tissue from the colon or from the breast or from the lungs biopsies, and you can give the specific drugs for the specific patient.
That's that's another interesting. I think one of the hospitals in New York, they're trying to do this kind of tailored medicine.
Yeah, they do. Yeah, they are. Yeah, we can do like if this in ASCP they will ask EGFR if there is a mutation in deletion mutation in exon. If there is an deletion, so there is a sensitive of for the drugs. that is tyrosine kinase inhibitors, right? If very sensitive, if there is a missense mutation in exon 21, then they will be resistant for this drug.
That's
This all we can do by personalized, medicine.
that's interesting.
Yeah.
Anybody has any questions? If not, thank you so much. Our next, yes, please. Tell.
Sir, shall I ask a question, sir?
No problem.
Sir, suppose if you find a variant by
Okay.
NGS analysis, will you
Okay.
it confirm it by Sanger sequencing? Because what I understand that
Not
as the sequencing, the gold standard is Sanger sequencing. Is it correct?
Yes, if that, see, not all the cases, if that reads is very low, if the reads of that specific variant which you identify and which match the phenotype, if the read is like 20x or 25x 30x, then in that case we suggest to do Sanger sequencing. If the read is very high like 100x or 120x, then no need to do Sanger sequencing. Okay.
I can add a little bit. There has like in USA, it's little bit. I think they made it complex. That is like different states they follow this kind of things like what Dr. Muit said. But New York State,
Yes.
they're so like they make the standard so high that they according to New York State, after doing the NGS, any lab need to do the Sanger sequencing. It's mandatory. That's why.
Yeah, it's all different. It's all depend on the lab to lab or
Yeah,
policies what
like
they
the
have,
state policy. Yeah,
state policies what they have, because okay, if one or two mutation if you find, then you can do Sanger sequencing easily. But if you find more mutations like 8 to 10 mutation and you want to do Sanger sequencing, then it will be very costly effect, and you need to design primers, and it will have some companies they offer free of cost Sanger sequencing after having the after identifying the mutations by whole exome sequencing. They offer free of cost Sanger sequencing.
that's correct. Because
Yeah.
of why I have seen that, like in New York Genome Center, they got samples from every state. So some of the state they
Yes.
can give the results very soon because they don't need to require the Sanger sequencing. But for New
Yes.
York state samples, and they have to do the
Yes.
Sanger sequencing.
And to know the segregation, to know the segregation analysis. Segregation means father mother need to be checked whether it's segregated or not. If, for example, you got a homozygous variant, okay, and one of the parent has also homozygous, then that variant is benign. Benign means not causative. Okay. For example, you got in case of autosomal dominant condition. Autosomal dominant means it is one allele is enough, one mutation, one heterozygous mutation enough to cause the disease. So if it is autosomal dominant in the child, has the disease, and it has a heterozygous mutation. Okay. Okay. Now the same heterozygous is present in both of the parent. It should be present actually one of the parent if it is autosomal dominant, not both. Okay. Then it is not causative. If one of the parent has the disease and having the same mutation, and there many things are there in genetics. You should know the expressivity of the disease. You should know there are lots of terms in the genetics like expressivity, penetrance. You know what is meant by penetrance, right? Penetrance. The many things are there. If you read that not Leela books, some of the Thompson and Thompson books for to know about all these terms, you can read that Thompson and Thompson about the expressivity, about the what do you mean by pleiotropy, what do you mean by penetrance. Because in breast cancer, BRCA1 is a germline mutation. Okay, one female has the breast cancer, but her mother has the mutation but doesn't has the breast cancer. That also can happen. That we call highly penetrant or low penetrance of the mutation.
Okay, sir, I have one question, but sorry, I just want to say thank you first to Dr. Boyd for such a very nice presentation. My question is regarding the profession of MLT. I'm sorry, I'm just going out of the topic.
No problem.
Just I want to ask that if now I have like me, I have the license as a technologist, MT. Um, sorry,
Hmm.
MB.
This
Yes.
no Abu, this answer
As
Abu. See, Adil work with us. Okay, Adil has Adil has done has cleared molecular technologist
MB.
MB, yeah, but he's in Saudi Arabia. Okay, he's working in molecular biology lab, working in, mostly on PCR. But now he want to shift to America. So what is the procedure to to
just
move to the
because
America?
just my family actually they are going there, and
Yeah,
my wife, yeah, and she's getting the postdoctorate in
postdoc. Yeah, he's getting
Indiana
postdoc,
state.
and he
Yeah.
wants to move there. So what is the procedure to get job there?
So,
This
like,
person.
So this is
obviously, like, if you are preparing for SMB, that could be one options. Like because highly skillful people has one options. There is another options. Like, if you have very nice research articles, you can apply through that. Or if you have like masters or PhD, like if you wanna come for PhD, that's one option. If you wanna come for postdoc, that is another option. If you want to apply suppose like SMP is very like only few people has that, and it's shows you have ability to direct one lab, like directorial ability. So this is another options to show you have one of the options, like you have one of the capabilities or potential. So this could be another options. I think I can like just I think you have my email address, right? Just email me. We can I can sit with you and discuss more because that would be another discussion.
Yeah.
yeah,
This discussion, because that is
yes,
another
yes.
discussion, because
Yeah.
uh, I don't know whether
I
it's difficult,
yeah,
whether it's
I
easy
can
to get job from here.
yeah, I think I can sit with you and discuss more with you. That would be I will be happy to share knowledge with you.
Sure. So thank you very much.
No
Well,
problem.
thank you. It's more time now, 2 hours almost.
Yeah, no problem. I am happy. I think some people had some other like schedules, and some of the peoples I know they work a night shift last night, so they could not join. But I think I can share this video if you give me the permission, Dr. Mohit, so that they can watch
Okay,
the
sure.
watch the discussions, and later if they have any questions, they can like email you, or they can
Yeah.
ask questions in the Discord or WhatsApp or Facebook
Yes.
group. That would be
Yes, actually, to crack the maybe to crack the ASCP, I don't feel you should have experience in all the field. It's not possible in your life, because you can't be experienced in all the field. You
that's
can't be
right.
experienced in microbiology. You can't be experienced in genetics. You can be. Only thing is, you should have we should have ideas in all
Yeah,
the field.
theoretical knowledge.
Theoretical knowledge at least.
Yeah, that's not realistic also. Our next group studies, I think Dr. Ramnam, she gonna share some knowledge on microbial
Yes, right.
portions. So I think it will be one informative also. Like
Tomorrow.
no, not tomorrow. Gave us time. I think I have to check the calendar. She gave us little bit of. I think it was 16 or 18, something like that. I'm gonna notify long earlier that the meeting. I have to also discuss with her what kind of topics she wanna cover, so that it will be nice also to get an idea of what kind of books or reference that people should check before joining. So.
Yes.
That's all. Thank you so much, Dr. Mohit, for your time. I think
Thank you, Abu.
we take a lot of time from you. Sorry for
Yes, no
that.
problem. Okay, no problem.
Okay.
Okay.
Thank you so much, and
Thank you.
have a nice weekend.
Thank you very much. Nice weekend.
Bye bye.
Bye bye.